Welcome to the Development in Drosophila module. In this talk, we're going to learn about cleavage in Drosophila. Drosophila is a protostome that belongs to the phylum Arthropoda. All insects and arachnids come under this phylum. In the case of Drosophila, the egg and the sperm fuse to form a zygote, and that zygote then undergoes the process of cleavage to form a multinucleated embryo. The cleavage is superficial cleavage, and hence it is a type of meriblastic cleavage. Once the multinucleated embryo is formed, a process of cellularization occurs to form the blastula. During the process of gastrulation, cell movements will occur, and we end up with a segmented embryo that will hatch from the egg to form a larva. The larva then forms a pupa, which can then undergo metamorphosis to form the adult fly. Let us first look at fertilization. In the case of Drosophila, the eggs are centrolecital, which means they do have yolk, and the yolk is in the center of the egg. What is interesting is that Drosophila eggs are activated prior to fertilization, which is different than what we have learned so far. And to make things even more interesting, in the case of Drosophila, the anterior posterior as well as the dorsal ventral axis of the embryo are specified prior to fertilization. Now, the sperm thus enters the eggs that are already activated, and this is because the calcium channels are activated before fertilization has even happened. Now, the sperms can enter into the egg through only one side, which is called as the micropyle. And when we look at the figure, the part in the yellow box, which shows that pointed structure, is the micropyle. And so that is where the sperm will enter into the egg, and the micropyle is actually located at the future dorsal anterior region of the egg. So where the sperm enters, that portion is going to become the dorsal anterior region of the embryo. Once fertilization occurs, the process of cleavage begins. In the case of Drosophila, and we had seen this in the past in the earlier chapters, that the nuclei divide many times without cytokinesis. And so what we have is a syncytium, which is basically one large cell with many nuclei. In the case of Drosophila, the nuclei will undergo division 13 times, so karyokinesis occurs 13 times. At the 9th nuclear division, of all the nuclei that have been made, 5 of them will migrate to the posterior end of the embryo to form the pole cells. And these are the cells that will produce gametes in the adult fruit fly. At the 10th nuclear division, the other nuclei that are present in the middle of the syncytium migrate to the periphery or the cortex of the cell and undergo division. And this kind of a structure is called as a syncytial blastoderm. So when we look at the figure that's there to the right, we're actually seeing different karyokinesis that is happening, and the numbers denote the actual division number. So let's look at the 9th one, where here you can see all the white dots are right in the middle. But however, when we look at the 10th karyokinesis division, now we are actually seeing that the nuclei are moving or migrating to the periphery. And so we see them on the sides. And this happens 13 times. So the nuclei undergo division 13 times. And when we look at the 14th, that is when cytokinesis will start happening and actual cells will start being formed. So up till the 13th cycle, there are no cells, but many nuclei, and they form what's called as a syncytial blastoderm. Now, as I said, after the 13th division cycle, the cell membranes start forming. And so the membranes will fold inwards between the nuclei to form cells. And this process is called cellularization. Microfilaments and microtubules play a role in the process of cellularization. And thus, cleavage in the case of insects is considered as superficial cleavage. This is a type of meriblastic cleavage. Meriblastic cleavage, if you remember, occurs in cells that have a lot of yolk in them. So in the case of superficial cleavage, large mass of central yolk confines the cleavage to the cytoplasmic rim, and that is what is shown in the figure. Blastoderm cellularization occurs in two phases. In the first phase, there is invagination of cell membranes between the nuclei, and furrow canals are formed till they pass the level of nuclei. When we look to the picture that is present on the right side, the parts that are in the black boxes show the furrow canals. Now, what should be noted is the nuclei tend to elongate during this process, and we can also see that in the picture to the left, where the nuclei are stained in red, the tubulin is in green and the actin is in blue. So as the furrow canals become longer and longer, the nuclei also start elongating. In the next phase, the actin protein that's present in the cells begins to constrict at the basal end, and that is what is shown in the small black boxes in the figure to the right. In fact, what will happen is these constrictions will then start becoming wider and wider until they join, and now you have one cell. And so this happens to all the nuclei that is present in that blastoderm. Now, when we look at the early development in Drosophila, it is directed by maternal proteins and mRNA that were placed in the egg during oogenesis. Now, till the nuclear division 10, the maternal proteins and messenger RNA aid in embryonic development. Around division 11 is when zygotic gene transcription will actually begin. And by the time we are at division 14, cellularization will occur at division 14, but it is also accompanied by a slowdown in nuclear division, an enhancement in zygotic gene transcription, as well as degradation of maternal mRNAs. And this stage is called as the mid-blastula transition. Thus, to summarize, we learned about the cleavage process in Drosophila, which includes a cellularization event and the mid-blastula transition.